So for, as Camilla indicated, for the next 20 minutes or so, I will talk about the lipid nanoparticle technology and how that enables us to deliver all kinds of nucleic acid therapeutics from small to larger. And as Camilla also said, I'm in Eindhoven, which is in the south of the Netherlands, where since 2019, I'm an assistant professor working on nanoparticle platform technology for delivery of nucleic acid therapeutics to all sorts of cells and tissues. And I will highlight just a few important technological breakthroughs for lipid nanoparticles, and I will give a few examples of therapeutic applications that haven't been mentioned yet by the previous speaker. So let's get going, right? I think Camilla also touched upon this, and Steve, of course, in the first talk, really about the final hurdle and probably the biggest bottleneck. But of course, if we want to use nucleic acid therapeutics and systemically administer them, we all know, and I'm pretty sure everyone that's attending this meeting is well aware of all the barriers. So I'll quickly go through this. Upon injection, of course, you want your nucleic acid therapeutics to stay stable in the circulation. So you need to avoid renal filtration, phagocytosis by immune cells, and degradation by nucleases. Of course, then you need to, be able to transport your therapeutic, transport it across the vascular and epithelial barrier, diffuse through the extracellular matrix, make sure it gets taken up in the cell, and then of course the big challenge, the endosomal escape, as Steve so eloquently talked about in the beginning of this session, and of course then unpackage the nucleic acid therapeutic itself, where it can then induce its either gene silencing function, gene expression, or gene editing function. So next to the systems that have been discussed, like the GalNAc siRNA conjugates or the antisense oligonucleotides, lipid nanoparticles is one of the other advanced technologies, and I'm sure you're aware of Onpattro, that was mentioned by Camilla in her lecture, the first lipid nanoparticle siRNA drug, or the first siRNA drug that came out in 2018. And of course, the two COVID-19 vaccines, basically the same lipid nanoparticle, which you can see a schematic in the middle of the slide, containing mRNA encoding for the spike protein as a vaccination purpose. So the LNPs are the big advantage of lipid nanoparticles are their modularity. Their interchangeability of the nucleic acid payload, so you can facilitate the incorporation of siRNAs, but also of much larger nucleic acids, mRNA, but also the components for gene editing. So it's really a platform technology that you can use to induce gene silencing, gene expression, or gene editing. So what does the technology consist of? Basically, there are four main lipids that are usually being used. The phospholipid and the cholesterol that make up the particle and just ensure that there's a stable particle. You have the PEG lipid on the outside that mainly plays a role in particle formation, the size, but also, of course, the particle stability once it's been produced. I will go into the PEG lipid a little bit more further along the line. And of course, the ionizable cationic lipid. So the ionizable cationic lipid is key. Ionizable because it can be charged at a low pH, uncharged at physiological pH. So of course, when you formulate these particles at a low pH, they efficiently complex the nucleic acid. When you inject them, you have a net near neutral charge on the particle, ensuring that it can circulate. And of course, once it enters the cell in the endosome where you have a lower pH, it's charged again, and it facilitates interactions with the phospholipids of the endosomal membrane, and aiding or promoting endosomal escape. And of course, because these particles are usually around 50 nanometers, they are very efficiently being taken up by the liver, as they can pass the defenestrated tissues, and they get efficiently taken up by hepatocytes. We'll go into that a little bit more as well. So why is the ionizable cationic lipid so important? Well, as you can see on the slide here on the left, is that in isolation, cationic lipids and anionic lipids present in the endosome, they adopt a cylindrical molecular shape. And that results in sort of packing in a bilayer conformation, which you can see here. Now, when those lipids are mixed together, cationic and anionic lipids, when they're mixed together, they combine to form ion pairs, adopting a molecular cone shape. And this promotes inverted non-bilayer phases that do not support the bilayer structure. And those are associated with membrane fusion and membrane disruption. And actually, this molecular shape hypothesis was used as a guiding principle for lipid design. So here you see, let's say, first to later on generations of ionizable cationic lipids, and as Camilla mentioned, these are the ones that were developed at the West Coast. I'm not talking about the lipidoids here that were developed by Dan Anderson and Bob Langer. Also very potent materials to deliver nucleic acids. But for sake of time, I'm just going to dive into the different aspects of these ionizable cationic lipids. So one of the first tested ionizable cationic lipids, called DOTMA, deoleal dimethyl aminopropane, it contained oleal lipid tails, so C18, conjugated to the dimethyl aminopropyl head group through ether linkers, and using again the molecular shape hypothesis as a guiding principle. The three components of these lipids, so we're talking about the head group and the linker, and of course the tails, they were systematically studied to determine optimal characteristics for each. So the molecular shape hypothesis really describes the macrostructure obtained upon hydration of a lipid with specific geometries. So more specifically, lipids containing tails with larger cross-sectional areas than the lipid head groups, they result in these H2 phases, or inverted micelles. And when the cross-sectional area of the lipid tails is smaller than that of the head group, this results in a cylindrical geometry, and these lipids tend to form bilayers. So comparing different lipid tail unsaturation suggested that the linoleal chains, those here on D-lin DMA, provide optimal particle internalization and potential to generate membrane destabilizing H2 phases. And so they first looked at the tails, and then they started looking at the head groups and the linkers. So the head group modified lipids were tested. A group of head group modified lipids were tested, and this led to the development of D-lin KC2 DMA. And this was actually the first lipid that Alnylam took forward for the Onpattro formulations. But a further screen and a further modification actually yielded MC3. And they actually stopped their clinical program and changed the formulation to contain MC3, which is now in the Onpattro formulation. And of course, on the right, you can see the sort of the next generation ionizable cationic lipids that are being used in both the BioNTech mRNA vaccine and the Moderna mRNA vaccine, you can see, of course, lots of similarities. But you see one of the big features is the introduction of ester linkages in the lipid tails, and that leads to improved tolerability and higher endosomal escape efficiency. And these lipids are more potent than MC3-based formulations following intramuscular injection. So that's just a brief overview of the development of ionizable cationic lipids. Now, how did they screen, let's say, at least until the MC3 formulation or the MC3 lipid? They used the factor 7 mice for that. The factor 7 mice contains of an intravenous injection of lipid nanoparticles containing siRNA against factor 7, which is a coagulation factor being produced in the liver. After 24 hours, you take blood and you determine the factor 7 levels. So the more efficient the LNP and subsequent factor 7 knockdown, the less factor 7 there is in the circulation. And the measure for that is called the ED50 of factor 7. So the effective dose whereby factor 7, 50% of factor 7 disappears from the circulation. So the lower the ED50, the more efficient the silencing and thus the lipid. So here on the left, you can see a plot of in vivo hepatic gene silencing activity, so expressed as the ED50, versus the pKa, the apparent pKa of lipids. And this was done, of course, in mice. And you see that the most active compounds, they exhibit an optimal pKa between 6.2 and 6.5. And if you plot this again in blue here on the right-hand side, so again, the hepatic gene silencing activity in mice versus the pKa and the calculated protonated and unprotonated aminolipids, or the ionizable cationic lipids in mole fractions. So the mole percentage of each ionizable cationic lipid estimated to carry a positive charge in blood is shown in red, assuming a physiological pH of 7.4. And the mole percentage of the ionizable cationic lipid estimated to be uncharged in acidified endosomes at a pH 5.5 is shown here in green. And you see that there's a sweet spot between 6.2 and 6.5. So this makes sense that the most potent lipids exhibit a pKa around that range. Mind you, this is really that was optimized for hepatic gene silencing. Just to give you an indication how this really enabled LNPs to translate to the clinic, is that you see that the initial LNP systems composed of DLIN-DAP, so all the way here on the right, they exhibited an ED50 of 40 to 50 milligrams per kilogram, and this is in mice. While the ED50 for MC3-based LNPs, so all the way to the left, they had an ED50 of 0.005 milligrams per kilogram. And that represents an improvement of more than 3 orders of magnitude with no increase in toxicity, and corresponding to a therapeutic index improvement greater than 8,000-fold. And this really helped the clinical utility of these systems. As I mentioned before, the PEG lipid also plays a major role in the efficacy of these LNPs. So traditional liposomes that you see here on the top right, so lipid bilayer surrounding an aqueous core, they usually have PEG DSPE or PEG 2000 DSPE, so C18 tails, to form a protective hydrophilic PEG corona to shield interactions with cells and proteins in the blood, and then you get an increased circulation time and target tissue accumulation, such as in the tumor. Now, the LNP systems, both Onpattro and the mRNA vaccines, they actually contain PEG DMG or something that looks very similar to that. It has C14 tails and it's considered a diffusible PEG lipid because it dissociates from the particle in the presence of a lipid sink, for example, when you inject it in the blood. And this really facilitates LNP uptake by cells, by hepatocytes. And the way that works, it looks like this. First, you get a dissociation of the PEG lipids from the nanoparticle, where there's a coding of the apolipoprotein E, which comes from LDL or HDL. And once this particle with this corona of ApoE passes the fenestrated endothelium, it can bind to the LDL receptor or other ApoE binding receptors on hepatocytes. There's very efficient internalization of the LNPs via endocytosis, and then you get the endosomal escape, as we talked about before, after which either the siRNA or the mRNA is released and can exhibit its function. Now, there is some evidence for the role of ApoE and the LDL receptor for this mechanism that I just explained. So in this graph on the left, you can see in wild type mice, so here all the way on the left, that lipid nanoparticles are very capable of reducing the mRNA and serum protein levels of factor 7. In ApoE knockout mice, you do not see this effect. However, so the effect is completely diminished in ApoE knockout mice. However, you can rescue that by supplying or adding ApoE to these lipid nanoparticles. So that shows that ApoE definitely plays a role in the efficacy of these particles. Secondly, similar results were shown in LDL receptor knockout mice. So again, although here in black wild type mice and in gray LDL receptor knockout mice, you see that the knockdown of factor 7 in wild type mice using LNPs is much more pronounced than in these LDL receptor knockout mice. So I'm not saying that this is the only mechanism by which these particles are being taken up, but it definitely plays a role, this sort of endogenous targeting system using the biomolecular corona surrounding these particles. So just to finish off, a few slides on applications of lipid nanoparticle drugs, and Camilla already discussed Onpattro. So gene silencing in the liver came out in 2018, a big breakthrough for the entire field. And she also showed these results from the clinical trial, basically showing the neuropathy impairment score, where the placebo group has progressive disease, and patients being treated every 3 weeks with an injection or an infusion of Onpattro, it would stabilize the disease because it inhibits the protein production that causes this fibril formation in these patients. Now, if you take lipid nanoparticles containing mRNA and you inject them intravenously, they also, of course, end up in the liver, but you can then use the liver as a bioreactor. And here I show you an example of already from a few years ago, where the liver was used as a bioreactor to produce antibodies against HIV. In mice, what you see here is the production of the VRC01 antibody, which is the antibody against HIV. And you can clearly see that if you give certain amounts of the mRNA lipid nanoparticles, you can detect antibody levels in mice 24 hours after administration, comparable or even higher to when you would just give the antibody. Now, importantly, if you challenge these humanized mice with SF162, which is a version of the HIV virus, you can see that in the higher doses of these lipid nanoparticles that were given before, it completely protects them from viral infection to a similar level as giving the antibodies itself. So that's really a nice strategy for this, but you can also think of enzyme replacement therapy, for example, to treat people with hemophilia or metabolic disorders. Just to briefly highlight, we all probably know this data, but just to show one of the strengths, of course, of a lipid nanoparticle mRNA platform is as soon as you have let's say a genetic code of something that you want to treat, the development can go very fast. And this is exactly what happened last year for the development of the COVID-19 mRNA vaccines. As soon as the SARS-CoV-2 genetic sequence was released, already, I guess, a couple of months after a clinical trial started, and there was a resulting in the emergency approval by the end of the year. Of course, based on these results that you see here for the Pfizer and the Moderna vaccines. I've had my first shot. I'm getting my second, I think, in a week or two. Now, I only put this in because it came out very recently, but these lipid nanoparticle systems are also now allowing or enabling gene editing following systemic administration. And there were 2 papers in Nature Biotechnology showing this in non-human primates. So a single guide RNA and mRNA encoding a CRISPR base editor were packaged in lipid nanoparticles and administered to macaques. And after uptake by hepatocytes, the CRISPR base editor introduces a loss of function mutation in the disease-causing gene PCSK9 and reducing PCSK9 protein levels and LDL cholesterol particles in the blood. So this in vivo base editing of PCSK9 in monkeys really paves the way for precise genome editing in patients. And you can see some of the results here. So you see the editing in the monkeys. And so this is the second study that was, I think, published actually in Nature. And maybe more importantly, here on the right top and on the right bottom, you see the change in levels of PCSK9 on the top and the LDL cholesterol in the bottom in the blood of those 6 monkeys. So 2 controls versus the 4 treated. And you see that the effect of the lowered cholesterol holds up, I think, around 240 days after 1 treatment. So that's really impressive. And just a few weeks ago, the results from Intellia Therapeutics, where they showed TTR, so the same gene as Onpattro is targeting with siRNA, they show that with a single administration, and I won't go through the details here, but what you can see is that their treatment actually led to a really, so these I think are 3 patients, so in total 6. You see a clear reduction of the serum TTR levels at day 28 following the dosing with the LNP treatment. It's too early to say something if this really does something to their disease progression, but these are similar levels that are being obtained with Onpattro. So there's a good indication there. And of course, having big implications for patients that are now being treated maybe every 3 weeks with an infusion, and maybe some side effects related from that can maybe be cured with a single shot. So I'll stop here because I already went a few minutes over time. I just want to mention that next week, of course, is the Controlled Release Society meeting, and then there's on July 28th. There's actually a workshop, if you want to know more about lipid nanoparticle technology, with speakers from both academia and industry. So Peter Cullis will give a talk there, but also people from Moderna, BioNTech. There will be some fundamental LNP work being discussed there, and some of the state of the art from the industry. So with that, I'll